All right, everyone. Thank you, everyone, for joining us tonight. It's 7:30 here at Central Time, so we'll go ahead and get started here. Super excited to moderate today's lecture. This is the National Fellow Online Lecture Series. This is a weekly installment. I'm Alberto Oseguera. I'm an assistant professor in family medicine and sports medicine here at UT Health San Antonio. I have the great privilege to welcome two amazing speakers, Dr. Fedoruk and Dr. Smith, today, who will be talking about performance-enhancing drugs. Just a few housekeeping before some introductions. Next week, we will have our, again, weekly fellow series. Will be the MRI series. We'll be talking a little bit about the shoulder and elbow. That will be given by Dr. Tate Greditzer 1 week from today at the same time, moderated by Dr. Robinson. Some goals, just as a reminder, this is an adjunct to everyone's home programs, educational content. This is really to provide fellows with direct access to educational experiences with AMSSM members and at times invited guest experts, also to assist in CAQ exam preparation. Some reminders: please mute your device's microphone and turn off your video. Please submit any questions you have through the chat function. I'll kind of sift through that and ask any questions at the end of the presentation. At the end of the lecture, you will get an evaluation, and please be sure to evaluate the lecture so we kind of know what to improve on and what you guys thought of today's lecture. Lastly, one of our new efforts is to facilitate more input from our audience. So there's a couple ways to do this. First, there's a web link that can be found by going to amsssm.org. You click the education tab, and then there's a fellow online lecture series link. The link is kind of to the top of the page as seen on this screenshot here. You can also do a scan by scanning this QR code. When filling out the form, we ask that you please keep in mind that the primary goal of the series is to assist in CAQ preparation for all sports medicine fellows. So without further ado, I'd like to welcome both Dr. Fedoruk and Dr. Smith. Dr. Smith is an associate professor of clinical family medicine at Georgetown University School of Medicine. She is a graduate of the University of Texas at Austin. She did medical school at Stony Brook School of Medicine, as well as obtained her MPH at Johns Hopkins Bloomberg School of Public Health. She completed her residency in family medicine at Christus Santa Rosa in San Antonio and a fellowship in community health leadership development at Georgetown University in partnership with Unity Healthcare. Along with Dr. Smith, we also have Dr. Fedoruk. Dr. Fedoruk is the Chief Science Officer leading the science and research team at the U.S. Anti-Doping Agency, also known as the USDA, with more than 20 years of experience in anti-doping and a PhD in pathology and laboratory medicine. Dr. Fedoruk also serves on many international expert committees and groups to advance the science of clean sport. Dr. Fedoruk has also served with the International Paralympic Committee and attended 7 Paralympic Games. He also serves as co-chair of the Scientific Advisory Board for the Partnership of Clean Competition, an important anti-doping research granting organization funded by the USADA, the major professional sports organizations, and the USOPC. So without further ado, I'll let you guys get started with the lecture. You guys can go ahead and share your slides if you have it to pull up here.
So thanks for the kind invitation, and pleasure to be here with Dr. Smith. Worked a lot together over the years, and we're going to start off with a little Q&A just to get to know us a little better, but really pleased and thanks again for the invitation to be back with the AMSSM. So I'll start off, and can you tell us a little bit about your career as an athlete, Tobie?
All right. Well, hi. First, everyone, thanks for inviting me. I'm excited for this hour and to work with Matt again. So I was a swimmer in Texas. I swam at University of Texas. and was a member of the national team for off and on through the 90s, 2000, and was NCAA champion in the mile, missed the Olympic team by a couple spots, a couple times. Try not to talk about that too much. But then did open water swimming and was world championships in 98, so a while back, in the 25K, and went on to do some other swims, English Channel, Manhattan. Trying to make a comeback and do a swim, but it's going slowly now. So not as much swimming now, but trying.
Great. What stands out for you in your memory as an athlete in the USADA testing pool?
So a lot of beginning of my career was USADA was not there. It was there was still testing, so it was not as organized as it was now, as it is now. And I remember in '95, so that was before USADA, at World University Games. We were in Japan. I got a little stomach upset and asked the team doc, can I take this medicine? Or he was giving me something. I was like, can I take this? And they were doing testing there. And he was like, not really sure, but depends. You got to weigh the risks and benefits. If you're not feeling well, maybe it's fine. And it kind of really stood out to me. Like, shouldn't you know that? But there really wasn't those resources. And I continued swimming, 2003, and was more in the USADA and had more. I think there's a lot more resources now. And I was still back in the time where we had a fax in our whereabouts, and so a lot more technology, I think, helps with the athletes these days.
Yeah, for sure. We try to lessen the burden, for sure. How did you get involved with USADA after you became a physician?
So my plan was to do sports medicine. That's why I went to Christus in San Antonio to do a fellowship there, but found my passion in community health. So after I was done swimming, I was kind of in medicine, no more sports, and I was very lucky that USA Swimming called me up and said, "Hey, we need to nominate people for the NGB, positions on the USADA board. Would you be interested?" And I was like, absolutely. And then I get the call from Edwin Moses, which was, exciting to hopefully all of y'all on here know who that is, who was the board chair at the time. And so I spent 8 years with y'all and just finished, I guess, December. I'm still sad about it. It's an incredible 8 years, and hopefully you'll learn more about, you'll learn more about USADA and what an incredible organization, and how much they're doing to protect our athletes and fight for fair sport.
Thanks, Tobie. And as a physician that has an athlete perspective, what are some of the main recommendations you have for the listeners today?
Yeah, and I think we'll have some takeaways here in the slides. And this is just, as I precept residents, and you all know this as sports medicine docs, but one of the things that my residents that maybe aren't sports medicine, when somebody has an injury and they'll be like, well, just take a week or two off. And I just am harping on, for an athlete, as you all know, if I missed 3 days, I figured my season was done. So that's my biggest takeaway. And we'll get some takeaways for doping, but I think it's just, and even it comes into doping, really knowing the athlete, and as we'll talk about, the TUEs and what you're prescribing, and knowing, the context of that athlete sport and what level they're competing at.
Great, thanks.
All right, so Matt, I get to hear about you, and Matt's had a quite incredible career in anti-doping. So how did you get started in anti-doping?
I got started after my PhD. I worked briefly in the biotech industry, and I realized I didn't want to stay in the pharmaceutical world. And I thought, I grew up as a downhill ski racer in the Canadian Rockies and wanted to mold my interest in science and passion with science with sport. And so I was fortunate to become a postdoc on a World Anti-Doping Agency research grant looking at blood doping. And that led to being able to work with the organizing committee for the Vancouver 2010 Olympic and Paralympic Games. We had to build a laboratory in Vancouver to analyze about 5,000 samples at the time, and work with the Montreal laboratory. That's the only lab in Canada that's accredited to do anti-doping sample analysis. So that was an awesome journey. Met a lot of people along the way, and ended up eventually at USADA 15 years ago. So it's been an awesome journey.
Awesome. So what have you found to be the biggest challenges in anti-doping?
I think from a science perspective, we're getting just so sensitive with the analytical techniques and detecting prohibited substances in athlete samples. And recently, because of that sensitivity, and I'll talk about it in the lecture, but the differentiating between what's actually due to doping and what's just innocent environmental exposure is a really important problem that we're trying to tackle. It's a very substance specific problem. And really, the credibility of the anti-doping system is at stake if we don't get things right. We want to get to the truth, and we don't want innocent athlete to punish innocent athletes. So this is an area that I'm super passionate about and very interested in. And we certainly, I'll talk about some interesting cases that we've come across in the recent past, which I think everybody will be interested in.
Yeah, I think that's one of the things I'm most proud of USADA's work in protecting athletes and not harming innocent athletes. So appreciate your work on that. So would you say science is ahead or behind cheaters?
Yeah, it's a great question. And we did some surveying, and in the athlete's mind, I think it's a toss-up. I think we're, in some athletes' minds, we're keeping up, and others, we've got some work to do. Certainly what keeps me up at night is not only these contamination issues, but athletes are turning to more using prohibited substances that are actually synthetic replicates of endogenous substances that our bodies produce naturally. And the physicians in the audience will be familiar with testosterone, EPO, human growth hormone. These substances are challenging from an analytical standpoint to detect. And so making sure we're keeping up with developing better sensitivity, better longer term detection, figuring out what metabolites may be indicative of doping versus not doping. These are all really important research questions that we're continually trying to tackle.
So why should physicians care about anti-doping?
Yeah, I think physicians, from an athlete's perspective, physicians are highly trusted individuals that interact not with one athlete, but you have the potential to interact with dozens, hundreds, thousands of athletes over your careers. And they really look up to physicians as being informed and trustworthy. And so these opportunities to educate the physicians, health professionals, because they do have such a large impact on athletes is really critical in making sure athletes don't make mistakes and they're getting up-to-date information about their anti-doping responsibilities. It's not an easy world to navigate. We ask a lot of athletes, and the prohibited list is very complex. So it's a real pleasure to be able to, spend the time today to do more to educate this particular stakeholder group.
Great. Thanks, Matt. And I guess we can
Yeah,
started here.
let's get into it. So just a quick outline for everybody. We hope to talk a little bit about some details and information about some of the core aspects of anti-doping and how it intersects with the sports medicine professional world and what you need to know. The importance of scientific research. I'll highlight some of the really important work that we've done recently and around the world to try to tease out some of and solve some of the current issues. I'll talk about, in that context, doping and contamination. We'll talk a little bit about what's coming down the pipeline with new sample collection innovation. This is an area that USADA is really passionate about, and we're aiming to develop and roll out new techniques that makes the process less invasive for athletes. That includes blood, new ways of collecting smaller volumes of blood, and then just some examples along the way of some research that directly supports anti-doping cases that you might have heard about in the news or noteworthy cases. So from an overall standpoint, we'll focus mostly today around the system that's been devised as the global anti-doping system, of which the World Anti-Doping Agency is the global regulator and aims to harmonize the anti-doping rules across countries. And so the mechanism for that is the World Anti-Doping Code. And there are over 700 signatories now to the World Anti-Doping Code and program. So athletes, no matter where they travel in the country, in the world, they should know when they go to individual countries that the testing process should be similar no matter where they go, and the set of rules that's applied is similar. Under that code process are, of course, anti-doping organizations. So you saw it as one example of that, where the Congress recognized National Anti-Doping Agency in the United States, but there's similar organizations around the world. So in the UK, there's UK Anti-Doping, Sports Integrity. Australia would be an example of another one down in Australia, as well as programs that are run through the sports federations or major event organizers when an athlete travels to a major event. So at the intersection of all of that is the athlete, and we need to make sure that the process for athletes continues to be fair. The process is harmonized across the world as much as possible, and by no means is the process perfect, but we're really aiming to make sure from the standpoint of consistency that there's a common set of rules that's applied. Now, athletes can also compete or participate in non-WADA code signatory sports. So a good example of that is the NCAA or the professional sports leagues in the United States. They wouldn't necessarily, those athletes could fall under the World Anti-Doping Code, say if USADA tested them ahead of the Olympic Games or the Paralympic Games, but they also have their own standalone programs, and those may be modeled off of the World Anti-Doping Code, but are also, there's some differences due to things like collective bargaining. So what makes an effective anti-doping program? And you all have probably heard about some of these components, but it's really important to realize that absent one of these components, it really has a strong effect on the overall effectiveness of the program. So of course, testing, both in and out of competition testing, 365 days a year, is really important from a deterrent standpoint, that athletes don't know when they're going to be tested in order to increase the unpredictability of the testing process. We ask athletes to file whereabouts so we can actually plan testing ahead of time. So there's a big burden on athletes to tell us, depending on the level of the athlete, even down to filing a 1 hour window every single day so that we could test them within that 1 hour window if we wanted to. There's a huge focus on education and awareness to make sure that athletes are properly educated on the rules, as well as a fair and consistent results management process, which athletes are afforded a due process through the process of being able to say, analyze their B sample if they have an A sample that tests positive, being able to challenge a positive test if they wish. And there's also a process by which they can be, for example, represented through the process by an independent counsel if they wish. There's an intelligence and investigations component, so that we don't just rely on analytical positives, but we can also assert violations based on investigation. So this is a really important component, especially when it comes to this particular group of individuals, athlete support personnel, of which physicians and other health professionals fall under that, being able to hold not just athletes accountable, but athlete support personnel under the rules is really important. We obviously want to protect and encourage whistleblowers, and there's a process by which athletes can offer substantial assistance. They have information in order to bring forward and possibly reduce their sanction based on other knowledge of doping within their sport. Science and research, my passion, as well as the laboratories, which are specialized in order to analyze urine and blood samples for the over 500 prohibited substances and methods. And then there's a process of therapeutic use exemptions available to athletes if they actually need to use prohibited substances or methods, and getting permission in order to do that. And we have a drug reference team in order to aid athletes in that process. So as I mentioned, from an anti-doping rule violation standpoint, many people think the process applies only to athletes. And certainly there's violations such as the presence of a prohibited substance in an athlete's sample, the use or attempted use of that substance, evasion, refusal, or failing to submit to sample collection or whereabouts failures that are unique to the athlete population. But beyond that, athlete support personnel and staff also fall under the rules. And so these are violations based on things like trying to tamper with the process, possession of a prohibited substance or method, trafficking, administration or attempted administration to an athlete, complicity, prohibited association, and retaliation. So it's important to know that there's a whole, a very vast list of potential violations that we're responsible for. Turn that over to you, Tobie.
So this is some of the resources that USADA has. I mean, there's a lot of education on there. You can go to the website and find the water code. You can find the prohibited list, which is updated annually, and the policy and procedures around TUE. So great resource if you haven't checked out the USADA website, really any resource you need.
All right, some of you may have questions around, well, how does an athlete get tested? And just like in the medical field, in the anti-doping space, there's so many acronyms that sometimes it, causes athletes' heads to spin off with trying to understand who's who in the anti-doping space. So just for the audience here to explain it in basic terms, the World Anti-Doping Agency, as I mentioned, is the global regulator. The World Anti-Doping Agency doesn't actually test athletes, so that's an important distinction compared to testing agencies. So there's really two different types of testing entities. There's testing entities that are under the auspices of sports federations, and then there's national anti-doping organizations. So many sports federations can outsource their testing to a third party, which is called the International Testing Agency. They run over 70 programs now around the world and conduct about 40,000 sample collections over the course of every year. So an athlete may be tested by a tester under the authority of the ITA that has been contracted essentially or given the authority to test from a sports federation. There are some sports that choose to have their own independent integrity agencies. And so athletics for track and field is an example of that. There's the Athletics Integrity Unit. And another example would be the International Skating Union. They still run their own program, although maybe components of that are outsourced to the ITA. Then you have national anti-doping organizations like USADA, the French Anti-Doping Agency, AFLD, Sports Integrity Australia, and even in some countries that don't have the resources to set up their own national body, there may be regional anti-doping organizations. So a good example of that is the Caribbean regional anti-doping organizations. There's many small countries in the Caribbean that all come together under the umbrella of the Caribbean RADO, which does testing and education in that region. And then, of course, the International Olympic Committee outsources its testing now to the ITA during the Olympics. The IPC still runs their own testing program at the Paralympics under their own authority. And they also are the sports federation for a number of sports like para athletics and para swimming. And then, as I mentioned before, you've got the NCAA, Major League Baseball, the NFL, and other professional leagues that run programs independent to the World Anti-Doping Program. From the prohibited list standpoint, it's obviously very complex. It's growing all the time, but just in very high level terms, there are the substances and methods in red, which are prohibited at all times. There's substances in blue that are prohibited and categories that are prohibited only in competition. And then there's some examples of beta blockers as one example of categories that are prohibited only in particular sports. So things like anabolic agents, hormone and metabolic modulators, diuretics, beta-2 agonists, and peptide hormone and growth factors are all examples of substance categories that contain prohibited substances at all times. You've got intravenous infusions, gene doping, blood manipulation, and non-approved substances. This is a broad class of compounds that include substances that are not approved by any regulatory body that have been that have the potential to be performance enhancing. And then you've got stimulants, narcotics, glucocorticoids, and cannabinoids falling under that in competition only prohibited category set. There's a global network of laboratories by which we send samples for analysis. So these labs are accredited by the World Anti-Doping Agency, and they're required to follow the international standard for laboratories. They're ISO 17025 certified, and they also are required to follow many different technical standards, technical letters, and methodology in order to make sure that the methods that samples are analyzed under are consistent from one lab to another. Right now, there's about 30 odd laboratories around the world, and we're fortunate in the US to have 2 WADA-accredited labs, one in Salt Lake City and the other at UCLA in Los Angeles.
So here's some more resources. There's a very large education component from USADA. So here again, you know, these ones are really geared toward the athlete and for training. The global DRO, and Matt's going to talk a little bit more about that when he goes over the TUE, but that's a resource from USADA. I think it's upkept by USADA and a couple other NATOs, UK, Canada, Swiss, some other. But it's basically, you can look up any medications prescription and OTC to see, is it on the banned list? So it's a really great resource that we'll talk a bit more about. Supplement Connect does not include supplements. Supplement Connect is another resource that is more about education and really trying to let the athletes know the risks associated with supplements. And then Health Pro Advantage over there is a CME program out of Stanford. I think it's like 2.5 CME. So really, really great resource that goes through particularly what we need to know as physicians around the TUE process. And so really more super clinical focus, highly recommend that resource. I think it was just updated this past month. It's a new one, so definitely recommend. Next slide.
So from the standpoint of how the process works around getting an actual therapeutic use exemption, it really depends on the level of athlete. And so for the higher level athletes that are part of a registered testing pool, these are generally international level and higher national level, for example, Team USA athletes. They can apply for a TUE directly with USADA after checking whether a substance is prohibited or not, and they receive a decision after an evaluation by an independent TUE committee, and they could either get the substance approved, denied. If it's denied, they get explanation as to why it doesn't meet the international standard for therapeutic use exemption criteria. And we could ask for more information, which is actually quite common. Sometimes applications are incomplete or the TUEC doesn't have enough information to make a proper decision, so we may come back to an athlete. We also have a process by which an athlete that's not in the testing pool can actually fill out a pre-check form to determine whether they need a TUE in advance. And depending on the level of the athlete, there may be specific categories of substances that an athlete does not need a TUE before they're tested. And so USADA will respond to these inquiries, can ask an athlete more information directly. Of course, all this inquiry is confidential, and an athlete may be told that they need to apply for a TUE or that no TUE is necessary at this time. from the TUE standpoint, really it's a multi-step process. An athlete needs to determine, and we provide the resources for them to determine whether they need a therapeutic use exemption. And so from this standpoint, really determining the status of the medication, whether it's prohibited or permitted, determining whether what competition status that they're under, and then doing that TUE pre-check to determine if indeed they need a TUE in advance. The first step, as Tobie mentioned, is to use Global DRO to determine whether the substance, the ingredient, or the brand medication, you can search either for many countries, are actually prohibited or not, and if there's a TUE, a special TUE process by which if an athlete requires emergency treatment, that that emergency treatment should never be withheld, even if it's a prohibited substance, and there's a process by which a retroactive TUE could be granted if the athlete can actually support through proper medical documentation that that emergency treatment with the prohibited substance or method was necessary. So you get your results around whether the particular substance is prohibited in competition or out of competition. There's a lot of detail here, but you can find this on our website as well, so you don't have to memorize it today, but then we give some detailed instructions as to what the next step in the process is, depending on the status of the particular substance. As I mentioned, that TUE pre-check form, it takes about 3 to 5 business days to get an answer back from the team. Often they get that answer back more quickly, but that's a really important resource to make sure that athletes get the correct answer. Obviously, physicians can use this resource as well. All right. Once you determine that a TUE is necessary, there's some additional resources that are available for specific diagnoses, which are particularly helpful. We've created checklists that are available, as WADA's also produced guidelines in order to make sure that the components of the diagnosis and treatment plan are clearly articulated, and that those can be provided as part of the TUE application. So here you see just an example of some of the areas such as ADHD, anaphylaxis, cardiovascular conditions, growth hormone deficiency. These are the type of categories where we offer some additional resources to be able to make sure that a TUE application is complete. We also, the second step of the process is to actually download the application, and you can go through it with an athlete in order to make sure it's complete. There's some categories and parts of the application that should be completed by the physician and signed off on by the physician to make sure that we're collecting a complete application. We do run programs for other sports that are not WADA code signatories, so the application can differ depending on if the athlete competes in those particular sports. And the third step of the process is to actually submit the application, a complete application. We've got a portal to be able to do that. That portal allows you to actually upload files to a secure website that USADA can then download the files. We used to, receive files by mail. You can still do that, but it's a little slow. So we've developed some online portals that allow for a more fast, increased speed in submitting applications. All right, so what does the TUE process look like from a timeline perspective? We've got the application submitted. USADA will confirm receipt within 3 days, 3 business days of submission. We assess that application and can get back to an athlete generally within 5 business days to determine whether that application is complete. We then send it out to the TUE committee for review. And we've been reviewing applications generally within a week to 10 days, but under the rules, it can take up to 21 days to receive a decision, but generally that is much more expedited than that 21-day process. And then do TUEs expire? They can expire definitely depending on the particular medication and how it's prescribed. So a TUE may be good for only a one-time use, such as an IV, or it could be as long as 10 years in the case of insulin and it being prescribed as a lifelong chronic condition such as diabetes. So it really does vary based on the particular athlete case and the circumstances.
I think we want to be really aware that there's a lot of medications that we use a lot. We're not thinking them as performance enhancing that are prohibited. So, certainly in our young athletes, acne, spironolactone. For asthma, there are some that are prohibited. Ones that are not are albuterol, although I think on the water list it's salbutamol, which is more international name for it. So it's also, there's a limit on how much you can use for that. So be sure you're checking on that. Insulin. And then methods, IV infusions. So 2018, you might have heard about Ryan Lochte that posted on social media that he was getting an IV infusion. It wasn't a prohibited substance, but he got more than the volume allowed and was given a 14-month suspension. So, think about just when athletes dehydrate, they want to get IV fluids. So those methods are banned. And then this one, the Clocetabol cream, which was not really used here in the US, but there is a cream with an antibiotic, I think it's with neomycin. So athletes, we'll go to other countries or they might be traveling and things that we're not aware of that we don't have here. That's not FDA approved. We don't really tend to use it, but it is used for, dermatitis and other skin ulcers, but it has that steroid in it that's banned. I think it's a derivative of testosterone. So the other thing I want to put out here is these are not just elite athletes. Some now are masters athletes. There's some places that they're getting tested. So when we're thinking about athletes that say, hey, I'm going to go run a marathon or a bike race, we want to find out there's more and more of those that are having random testing as well. It's a lot of things we use, testosterone in older athletes. So side note, just kind of keeping in mind the all range of athletes that we're treating. So dietary supplements, obviously that's a big thing with our athletes. Definitely use it at your own risk. They are not, as we know, FDA approved. There's a lot of risk, strict liability. So it doesn't matter if the athlete says, I didn't know this was in there. It doesn't matter. They ingested that, it's in their system. There's certainly lots and lots of cases of contamination, mislabeling, contamination with substances that are prohibited. This Supplement Connect is a resource really more to educate about the risks. It's not one that will tell you, is this safe or not safe, but it does show a high risk list of supplements. And really, we need to be just educating athletes that is really not worth it. I mean, the training and nutrition take priority versus a risk of taking a supplement that you have no idea where, what the risks are with that as far as with doping. So this is the high risk list on there. And I don't know if Matt, interest of time, if you wanted to point any of those out or not, but this is an important one to look at, ones that are around weight loss and, recovery, but there's some in here that are really high risk for containing substances that are banned.
Yeah, unfortunately, it's a non-exhaustive list, but it's important to point out that USADA does, when we identify products that either contain a prohibited substance that's listed on the label or an athlete tests positive because of a prohibited supplement, we we have a list available that as a resource to be able to track those things. There's no way in the United States to know how many supplements are actually on the market. So as I mentioned, there's no way to have an exhaustive list, but definitely our stakeholders feel that this list is helpful at educating them around potentially high risk products. And then the only way to reduce your risk of testing positive, or an athlete's risk of testing positive from a supplement, is not to use a supplement. But we've recognized that that's not the reality for many athletes. They do choose to use supplements, and there may be reasons medically or nutritionally that a supplement is necessary. So what we recommend is that athletes choose the NSF Certified for Sport third-party testing program. This is an independent body that evaluates products directly from manufacturers and certifies them with the NSF Certified for Sport mark, and as analyzed for prohibited substances. It's not 100% foolproof. So as I said, the best way to reduce your risk to zero is not use supplements, but it's really a way to make sure that an athlete can reduce their risk, because there are so many unknowns with products that are available on the market and available for purchase, not only in stores, but online today. And then we see, unfortunately, a lot of false advertising. So this is where it's buyer beware. We see examples of where supplement companies have actually called products WADA approved or USADA approved, and we would never approve a specific product as safe for an athlete. So that's a really important point to make sure this group understands, that if an athlete comes to you and says, "Oh, it's USADA approved, I see a USADA mark on this," that is false advertising, and we would never approve such a product. So look for that NSF Certified for Sport label, and know that there are some unscrupulous companies out there that incorrectly use marks in order to market their products.
So just some takeaways from here is, being aware if your athletes are subject to drug testing, ask the athletes. And also, as I said, some of our masters athletes also are subject to testing for some of the masters competitions. Always check the Global DRO, be familiar with the TUR process, and that resource that I put in there goes through it as well. And there's also a resource on USADA that you can look. Counsel again on supplement and online pharmacy risks for sure. Athletes are getting these things mailed in from wherever, and listening to information, on social media about what they should be taking. And I don't think really understand how risky it is as far as from doping perspective. Really advise athletes on documenting all meds and supplements used. And Matt's going to give some examples of some innocent, some other cases where this was really important. So documenting, any over the counter, this is what I think we'll get into maybe the sunscreen one. So really has helped athletes get out of trouble and can show what things they've taken. And then consult USADA. There's lots of resource again on the website. And then again, the CME resource there. So Matt, I think in the interest of time, I want to make sure that you get to the passport and the dried blood spot testing and some of the really interesting, innocent, the cases that you went through. I don't know, make sure we get through some of that, because I know we're catching up on time here.
Thanks. I'll move through these fairly quickly. But one interesting development in anti-doping in the last 10 years has been the increased use of the athlete biological passport. And we recognize that there are intra and inter-individual differences in athletes. And when we look at anti-doping from a forensic standpoint, we're looking not only for the presence of prohibited substances, but we can actually follow biomarkers in an athlete's urine and blood samples that can be indicative of abnormal physiology that is due to doping. So really focusing on a new tool in our toolbox, using some advanced mathematics in order to try to predict what an athlete's normal physiological ranges may be for that particular individual, and not rely on population-based averages, has been particularly useful to guide things like strategic testing, as well as some of the analyses that are applied to substances such as EPO or testosterone that can't be applied to every sample because of resource limitations or other limitations. So the passport has become a really important tool to follow longitudinally an athlete's testing history and the biomarkers that are collected, in this example, testosterone over epitestosterone in urine samples, and really being able to tease out whether an athlete is or above or below their own individual variability that is tracked over time. And of course, this is reviewed by independent experts to make opinions on whether this is just due to normal fluctuations, due to things like exercise, diet, normal physiology, or medical conditions, or it could be due to doping. And there's quite an extensive process that we go through to evaluate every single sample in an athlete passport. Here's an example of what an abnormal passport might look like for blood doping. You see here what's known as an atypical passport finding, where we've got hemoglobin levels that are either above or below the athlete's intra-individual reference range that you see in red. As I mentioned, laboratory sensitivity is ever increasing, and so we've got a detection window of a particular substance and a limit of detection of that substance, but that detection window is ever increasing due to the lower limits of detection of the laboratories. And that causes some challenges that we've encountered in the last 5 to 10 years with particular substances that may appear due to environmental exposure. And so we've published in this area with things like not only dietary supplements, which obviously can be contaminated with prohibited substances, but now concerns around food such as meat contaminants in growth promoters in meat, and drug contamination or other metabolites that are produced by non-prohibited drugs that may be similar or the same as prohibited substances, as well as direct contact and causing a positive due to things like intimate contact with or contact with pet medication. These types of things are real, I wouldn't say common issues, but have come up in anti-doping cases, which have required quite an extensive amount of research to try to understand what might be due to doping and what may be innocent exposure. Athletes have significant anxiety around this. We did some surveying, and athletes, they do have significant risk about things like contamination due to supplements, 88%, risk due to pharmaceuticals, 73%, meat, 42%, environment, 34%, and intimate contact, 32%. So these are real issues that we're addressing because it causes athletes angst, and we want to make sure the system remains fair to athletes. So are all tests due to the positive tests due to the result of doping? Certainly prohibited substances can appear due to contamination. It requires the athlete's burden to show where those substances came from, and we want to eliminate the risk of falsely accusing athletes of doping through to no fault of their own. I really recognize that athletes are not scientists, and so we want to make sure that we understand whether these scenarios are plausible or not, because often athletes don't have the resources to understand, or in some cases even identify whether these contamination issues are even possible. But we want to minimize the risk of opening up loopholes for dopers and using these excuses in order to circumvent the rules. And this is all due to increases in lab sensitivity, and we need to balance that with the need to increase detection windows. And there is significant work going into things like identifying new metabolites that may be due to doping versus other metabolites that may only be present in cases of contamination. So that's very interesting work. As I mentioned, there's various areas that we're looking into and solutions. So things like we can solve these problems by application of thresholds or reporting levels so that samples that have low amounts aren't even reported out. Things like identifying metabolite patterns, like I mentioned, or application of specialized testing, such as the application of carbon isotopic ratio mass spectrometry that allows us to differentiate between, say, the synthetic use of a substance and the natural production of a substance. We've seen some innocent source positives, and so things like pharmaceutical contamination is more of a concern now, certainly after COVID and the implication of quality control and some breakdowns and outsourcing of pharmaceuticals more to third world countries. This is becoming an issue in the area of diuretic detection, meat contamination due to the use of growth promoters. There's been actually hundreds, if not thousands, of cases of clenbuterol contamination due to athletes traveling to places such as Mexico, China, Guatemala, areas where you may not have good agricultural controls on the use of growth promoters, dietary supplement contamination, intimate contact, as I mentioned, and other environmental contamination. We've published in the area of, for example, diuretic contamination of generic pharmaceuticals. We've seen over a dozen cases now of athletes using otherwise permitted medications and testing positive at low levels due to diuretics. And I'm actually really pleased to share that in this area, we've had the rules changed, and the way that laboratories report these low-level positives is now atypical findings, which leads to an investigation and not necessarily an adverse finding that would result in an immediate sanction. So the rules are evolving as we experience and come and find solutions to some of these real problems that we're seeing. Tobie mentioned online pharmacies. I just briefly wanted to mention the use of online pharmacies is increasing, and these pharmacies really circumvent the checks and balances that are now in place, the protections that are in place when athletes or others go to real brick and mortar pharmacies. So this is a real concern, and there's an increase in individual self-medicating or trying to find medications at a cheaper cost, and this extends into the sport world. So there's roughly 40,000 online pharmacies now, and 95% of them are operating illegally. So this is a real concern, and the trends are just due to things like shifting buyer behavior, technology-driven disruptions, price and access issues, and the rapidly growing phenomenon of consumerism in healthcare growing overall. And the concerning thing is that there's surveys indicating that healthcare professionals are not actually discussing these issues and these real world concerns with patients. So this is an area of awareness for you all to be able to communicate this, that this is actually not a smart tactic by athletes to shop online because they may not be getting what they necessarily think they're purchasing. From the standpoint of innocent source positive, I just wanted to quickly share a few cases. This is a case of a substance called caprimorelin. It's prohibited under peptides and hormones. And this is an instance of an athlete being exposed because they were treating their pet dog. This is an appetite, a drug that promotes appetite in sick animals. And this is a case where an athlete tested positive due to dermal exposure. And we were able to do some experiments demonstrating that even as little as a few microliters exposed on the skin can cause a urine positive. So this was a high profile athlete that avoided a sanction due to our work. So we were particularly proud of this particular case. And there was actually a subsequent other athlete in a similar situation just around the Paris Games that due to our research was able to be exonerated as well. Another area that's concerned that you should be aware of is the use of carbonic anhydrase inhibitors such as dorzolamide and brinzolamide. Athletes can be, it's not actually prohibited to use these through ophthalmic eye drops, but the problem is that there's been cases of which eye drops, athletes have been exposed to eye drops by treating either pets or being exposed through secondary exposure through individuals that are using eye drops and testing positive. And as you may know, these substances bind to red blood cells and can last a very long time in the human body. This is a sunscreen case that came up before Tokyo, where 2 athletes tested positive for a particular metabolite that was common to, and it was unknown at the time that this metabolite was common to a substance that was available in cosmetics as a preservative called chlorphenesin, and it was shared. It shared a common metabolite with a prohibited stimulant called meclofenoxate. So through some sleuthing work, we were able to determine that some common sunscreens that are available on the market were actually causing athletes to test positive due to some changes on the laboratory side in reporting this particular metabolite. So we were able to get the rules changed and sort this all out now so it doesn't happen anymore. But this was very concerning. Over 80 athletes were actually reported positive and through our work prevented from serving any sanction and exonerated. So this was particularly really important work. And the last case I'll share is around roxadustat. This is a HIF stabilizer used as an alternative to EPO. And this is a substance that isn't actually yet approved for use in the United States, or is recently approved in the US, wasn't approved and is recently approved in the United States, has been approved in Japan and some other European countries. But we had a case in which an athlete actually tested positive for this substance for over 2 years. So many times, we don't fully understand the excretion profile of new substances on the prohibited list. And this just illustrates that it's really important to do this work to understand that an athlete could test positive over an extended period of time. And obviously, for a prohibited substance like this, it becomes really important from a deterrent standpoint. One, our methods are so sensitive that it can detect down to almost individual picograms per milliliter. But we also need to understand to make sure that we're not unfairly accusing athletes multiple times because these substances can persist. And then to end it off, I wanted to talk about just new blood collection processes. So you all will be familiar with traditional venipuncture and the need to ship samples to a laboratory under cold chain shipping. This is typically small amounts of blood collected from athletes for analysis of growth hormone and blood parameters, but we are working on more athlete-friendly ways to collect blood. This is an example of an arm device that collects capillary blood from the upper arm that collects dried blood spots that can then be used for analysis of a whole host of prohibited substances. And we're also working on a new version of the device that collects small volumes up to 500 microliters that can be analyzed for a broader range of prohibited substances and that can be operated without the need for a phlebotomist in the field. We've tested this through different processes, such as the athlete biological passport, to show its equivalency to traditional blood draws, which is very exciting. And we can actually collect multiple tubes of blood depending on how it's applied and the application of this device. So that's really beneficial when there's things like an A and B sample that need to be collected. And finally, I'll end off by just the importance of research and innovation in the work that we do. At USADA, this is a really important hallmark of our overall program, and this has allowed us to better understand things like excretion, pharmacokinetics, and the need for new sample collection methodology. So I really want to thank everybody for their attention. You can reach out to me at any time. There's my contact information, and happy to maybe take a question before we end off today. Thanks.
Yeah, thank you. That was amazing. I think, we all could definitely learn a lot more when it comes to, anti-doping testing and, the science behind all of that. I will, there was a few questions in the chat. I will probably email some of the others to you guys. And if you can email me those responses, I can respond to the question that was given. But one of the questions I found was interesting was, do the national anti-doping agencies have a conflict of interest when it comes to positive tests for their own national athletes?
Yeah, great question. It's certainly come up in the context of some of the interesting cases globally. And I think it's really important to look at the overall governance of the entities in the US. We're a completely independent body, independent agency from any sport. So it's really important to have that arm's length to make sure there's no conflicts of interest. And in the case of even our board of directors, there's nobody serving on the board that are currently involved in sport, such as the USOPC or other sports bodies. So this is really essential to eliminate those conflicts. And certainly from an elite athlete standpoint, it's also important to point out that our athletes can not only be tested by USADA, but they can be tested by their International Sports Federation and other entities that may have jurisdiction over those athletes during international events. So it's not just USADA testing US athletes. All those tests are combined together on an individual athlete testing program. So that ensures that we're not just testing our own, and we also have jurisdiction over foreign athletes when they come to the United States.
Excellent. Well, just again, for the interest of time and for all those East Coasters out there, I know it's getting a little late. So for the other questions, I'll go ahead and send those to both Dr. Smith and Dr. Fedoruk, and I will send those replies to the people who asked those questions. So with that, thank you everybody for joining us tonight. I'd like to thank again Dr. Fedoruk and Dr. Smith for an excellent lecture, and hopefully you guys learned a lot tonight. I sure did. With that,
thanks again for the invitation. Thanks, everyone.
Yeah, thanks so much.